Hello, my name's Claire Green and I'm working for the Clinical Genetics Department here in Leicester. This podcast is about how clinicians determine if a missense variant is the cause of a disease in the family. To understand how clinicians determine the significance of a missense variant, it is helpful to know a little bit about genes and genetics. Genes are the chemical messages that build and maintain the body. They are passed down from generation to generation. We inherit one copy of each of our genes from both of our parents. This means that we have two copies of every gene. The genetic code is made up of four bases, known as A, C, T and G, which act as the genetic alphabet. This acts as the blueprint for the production of proteins. Each sequence of three bases codes for an amino acid. An alteration in the sequence of bases can change the amino acid produced. Amino acids join together to form complex structures, called proteins. Proteins are vital for a number of chemical reactions in the body and often do not function if the genetic code is altered. When the genetic code is altered in a subtle way, the protein is still produced, but it can be difficult to predict the effect on its functions. A variant which causes disease is called a pathogenic variant. It can be difficult to be 100% sure whether a variant is pathogenic or not. Clinicians use a number of techniques to understand the significance of these variants. Firstly, the clinical team will look at the published medical literature. They will look to see if the variant or variation in the gene has been described in patients who have the disease or in large unaffected groups. If the variant is present in patients who have the disease, it is likely that the variant is pathogenic. Secondly, it can also be helpful to read the medical literature to see if this variant is present or part of the gene varies in other organisms found in nature. Vital parts of genes have tended to remain the same through evolution. Therefore, if the variant has never been described in humans or other species, it is more likely to be pathogenic. Thirdly, we can offer to test other relatives with the disease to see if this variant has been inherited by other relatives with the condition. If it has not been inherited by other affected members of the family, the variant is likely to be insignificant. If the medical condition has occurred for the first time in the family, and the variant detected in the affected individuals is not present in other family members, this may also provide further clues to the clinician as to whether this variant is relevant. In the medical notes, we often state that the variant has arisen de-novo. De-novo means that the variant has occurred for the first time in the affected individual, and this often increases the likelihood that it is pathogenic. Fourthly, the chemical structure of the gene change may be important. This and the use of computer modelling will be described further in the next, more in-depth podcast, Understanding Your Missense Variant. Thanks for listening, and please do not hesitate to contact your clinical genetics centre if you require further information.